Here we are for chapter number 6, peptides and proteins. Peptide bond formation. A peptide bond is an amide linkage formed when an unshared electron pair in the amino nitrogen attacks the alpha carbonyl group of another amino acid in acyl substitution reaction. A water molecule is released. This is dehydration reaction or condensation reaction. And what you've got now is a linkage between two amino acids. This is a dipeptide when there are two amino acids, a tripeptide when you got three amino acids, and tetrapeptide and so on as the number of amino acids increases. So I've been using this terminology a little bit, but N-termini and C-termini, N-terminal amino acid and C-terminal amino acid. Your N-terminal residue is the one with the free amino group, and the C-terminal residue is the with the free carboxyl group. Peptides and proteins are read in an N to C direction. So this is effectively what we're looking at. We're looking at, when it comes to the formation of a dipeptide, we've got a reaction between the N-termini of one amino acid and the C-termini of another amino acid. The result is our condensation reaction, in which case a water molecule is released, and a linkage between the nitrogen and the carbon of, the carbonyl carbon of another amino acid. Now, peptide bonds are created within a living cell by ribosomes. These are artificially synthesized peptides, can also be made, however, you have some limitations on the size of that molecule. In terms of synthesis of proteins within a cell, this is effectively what you're looking at. An enzyme known as peptidal transferase is going to take those N and C termini of respective amino acids and form that peptide bond. Now, a peptide or a protein is read from the N termini to the C termini. So residue number 4 or something like that is R group number 4 after, let's see if we count up our peptide bonds. So this would be a peptide bond, this would be a peptide bond, and this would be a peptide bond. So it sounds a little bit weird, but if you have 3 peptide bonds, that means there are 4 amino acids within that molecule. Within that polypeptide chain. Now, peptide structure. Large polypeptides have well-defined three-dimensional structures. This is known as the native conformation, and it's derived directly from the specific amino acid sequence. So, which amino acid is at the N-termini all the way through to the C-termini? That's going to dictate what the 3D structure of that protein is. Now, most polypeptides spontaneously fold into a single biologically active form. This is work done by Linus Pauling, but also kind of confirmed by a guy named Christian Anfinsen. Now, these peptide bonds are unique because they have partial double bond character, because you've got delocalization of a bunch of electrons basically from the atoms that I'm circling here. You've got effectively this almost double bond type character resulting from all of those delocalized electrons around those atoms. Now, protein function and shape. Proteins can perform a ton of different tasks. They can be involved in catalysis, so they can convert reactants to products. They can also be involved in stress responses. Because of this diversity, proteins are going to be classified by their shape and their composition, so their amino acid composition, as well as something of their composition of their structure. Now, protein shape classification falls into 2 major categories, and those major categories are fibrous proteins and globular proteins. Fibrous proteins are generally going to be insoluble in protein, and they're a little bit more predictable and well-defined. They play structural and protective functions. For instance, keratin and collagen are both fibrous proteins. Globular proteins are a little bit different because they're very, very compact, for one thing that makes them different, but they're also water soluble and they have a much more dynamic function. Nearly all enzymes that we'll study later on, and when we talk about the metabolism portion of this class, they are all globular. So additional structures that proteins can kind of form, 3D structures, are these sorts of models that are shown below. Now, some of this is a little bit beyond where we are so far, but as you can see, the diversity of these structures, you've got some things that we'll just call little helices here. This one's almost all helices. This one's helices just like the first one, but these helices, they wrap into basically a ring. And so you've got some diversity amongst all of these like different folds. We've got helices that are seemingly sort of random, and then helices that are wrapped into a ring, and then, well, this protein right here doesn't really seem to have many helices, but instead it's got these little arrows that are kind of flat arrows that they maybe look like they're about a single plane. But we'll get into that a little bit later. At any rate, I'll see you in the next video.